So, moving on to the actual details of digestion, we need to know that it takes place in two stages: physical digestion and chemical digestion. Now, in your booklet, you've got these notes written down, so just ensure as you go through here that you've got an understanding of what's happening. Physical digestion. This is just breaking up large pieces of food into smaller pieces. It will start in the mouth with slicing and chewing with the action of teeth. One of the main reasons for this, aside from being able to swallow food, is that it increases the surface area for enzyme action, and that is the chemical digestion. It will continue in the stomach with the churning caused by the contraction of the muscles in the stomach wall. In chemical digestion, what you've got is actual chemical breakdown of food molecules into smaller soluble molecules by enzymes. So this is hydrolysis, and you all know about hydrolysis reactions from your biological molecules topic. So all digestive enzymes are hydrolases. They break chemical bonds by the addition of water. All enzymes, as always, are specific, and more than one enzyme is needed to hydrolyze a large molecule. One enzyme will hydrolyze a large molecule into sections, and then other enzymes will hydrolyze those sections into their monomers. So when you have really large complex molecules, there will be a few stages before we get down to the monomers. And we need to hydrolyze down to our monomers so that these smaller molecules can get absorbed into the bloodstream. So our three main groups of enzymes are carbohydrates, proteases, and lipases. Let's start with digestion of carbohydrates. So you can see here that this happens in two stages. Now this is where there's quite a difference between what you were taught at GCSE and what you need to know for A-level. If we take starch to start with, it's a polysaccharide. We have two types of amylase that can break this down into smaller molecules, hydrolyze it. Whilst I'm on this matter, whenever you're writing or answering questions on digestion, use the term hydrolyzes rather than breaks down. It is a more accurate terminology to use. So just get into the habit of using that. But here we have salivary amylase and pancreatic amylase that will hydrolyze starch into maltose, the disaccharide that, as you know, is made up of two alpha glucose molecules. We then have another enzyme called maltase that is made in the ileum and the epithelial cells, and that will then hydrolyze that disaccharide into glucose. So let's go over that. Salivary glands secrete salivary amylase. That amylase hydrolyzes starch to maltose. Food reaches the stomach. That amylase will be denatured. But when it passes into the duodenum, pancreatic juice contains the pancreatic amylase, so there's further hydrolysis. Both in the mouth and in the duodenum, there are salts that will maintain the pH at neutral for optimum conditions. Peristalsis moves food along the intestine. And then we have these membrane-bound enzymes. So we've got membrane-bound maltase that are on the epithelial cell membranes that will hydrolyze that maltose to alpha glucose. You need to pause the video now and fill out your notes in the student booklet. Membrane-bound enzymes are an important group of enzymes that, if you look at this diagram here, you can see these are the microvilli on the top of the epithelial cells that are lining the ileum. And you can see here that this is maltase. This is an enzyme that's bound to the membrane. It doesn't come out into the lumen of the ileum. It stays there. And the maltose molecules that are moving along here will then be able to form enzyme substrate complexes here, and then they're hydrolyzed into the glucose molecules. These can then be absorbed across those epithelial cells, and we'll talk about that in a moment. This diagram here shows exactly the same thing. It's a little bit more simplified. Here's and this uses the term disaccharide because we've got different disaccharide enzymes. But again, you've got that enzyme substrate complex is made here, the hydrolysis reaction takes place, and then we've got our monosaccharides. Okay, now for our specification you need to know that sucrase and lactase, that are also disaccharide enzymes and membrane bound, they will obviously hydrolyze sucrose into glucose and fructose, and lactose into glucose and galactose. Again, this links in really well with your biological molecules. Proteins also have that similar process where you have various enzymes that will hydrolyze a large protein molecule into smaller molecules and then eventually into the amino acids, which are the monomers. So if we look at the diagrams again here, we have 3 types of protein enzymes that you need to know about. They are all peptidases, but we have endopeptidase, exopeptidase, and dipeptidases. The endopeptidases will hydrolyze large protein molecules within the middle of the protein and break them up into small chains of amino acids. Then what you then end up is sort of polypeptides that have exopeptidases, and what they will do is they will work on the peptide bonds on terminal amino acids. So they'll work on terminal amino acids. These are secreted from the pancreas. Your endopeptidases are made either in the stomach or the pancreas, so we've got them made in 2 separate places. Okay. But then finally we have our dipeptidases that will hydrolyze a dipeptide into the constituent amino acids. And these, again, like the disaccharidases, are membrane bound. They're found in the ileum, and they will then form those enzyme substrate complexes on the epithelial cells. And that is the digestion of proteins. Lipids have a slightly different way of being digested. In your student booklets, you can see that the notes are there for you, so just let's work through this so that we've got a good understanding of what's going on. If we start with a large molecule, our lipids are large molecules, they form globules. They will be hydrolyzed by lipases, but before that occurs, the presence of bile will emulsify those large molecules and break them up into smaller globules that have a much greater surface area. What will then happen is these lipases will hydrolyze the ester bonds and they will form fatty acids and actually monoglycerides. So the monoglycerides, this is a diagram of one here. Here's your glycerol molecule with your 3 carbons, and it has 1 fatty acid chain on the end. So again, these lipases are produced in the pancreas. They hydrolyze that ester bond. Monoglyceride is the glycerol with 1 fatty acid attached. And the bile, beware, it is not an enzyme. So it's not doing any hydrolyzing, it's not doing any chemical breakdown, it's just emulsifying those large fat droplets into smaller droplets. These have a name, of course, because it's biology and we like to have a terminology for everything, called myceles, okay? And they're increasing the surface area of the lipids. Bile salts are made in the liver and stored in the gallbladder, and they'll come into that duodenum, that beginning part of the small intestine, via a little duct. Sorry, I'll go back one page. At this point, you need to go to your student booklet and you need to fill out the table that summarizes all the enzymes of digestion and where they're made. If you are not sure about any of the information, you need to go back and reread the slides or reread the textbook to ensure that you've got all that information. So let's move on to absorption. So this is a bit that's really important. This is where we're going to get those smaller molecules into our blood and then we can utilize them. So again, there's a lovely diagram here to show you what's happening inside the ileum. There's those folding, the folding of the wall, the villi here with the microvilli on the outside. And this is a diagram of a microvillus. So you can see the microvilli are here, and this is one villus, and you can see how that surface area will really increase. Now, you need to know the adaptations of the ileum and those adaptations and how we're adapted for the function of absorption. And simply put, we've got villi increase the surface area for diffusion. They're very thin walled, so they reduce that diffusion pathway, and this links into Fick's law. Muscle contraction maintains a diffusion gradient, so we're keeping that diffusion gradient going along the ileum. Remember that the ileum and the small intestine move things along with that peristaltic movement. Blood vessels carry away absorbed material as soon as it diffuses across into them, and so again we're maintaining a diffusion gradient. And lastly, we have those microvilli on each villus that increases the surface area for absorption. Now, again, so here we have another diagram that shows those epithelial cells with the microvilli on the outside of them. And here's a capillary, just to remember that actually the hepatic portal vein carries glucose to the liver. Most of those blood vessels will flow into this vein to the liver. Glucose is absorbed from the blood by cells for respiration, either aerobic or anaerobic. Excess glucose will be converted to fat. So at this point, I would urge you to pause the video and in your booklets have a go at writing out what is happening in terms of absorptions of amino acids and glucose co-transport. You've done this in the transport across membranes module, so this will be like a revision activity. Okay, so let's quickly recap what's happening here. First of all, we've got our lumen of the gut here. Whenever you see these diagrams, just familiarise yourself with where we are, and we've got blood capillaries here. Sodium ions are transported out of the epithelial cell via the sodium-potassium pump, okay, and that is down here. So sodium will be pumped out. This obviously requires ATP, so you'll find lots of mitochondria in these cells, and potassium comes in this way. This maintains a low concentration of sodium ions inside this epithelial cell compared to the lumen, and that's important because it sets up that concentration gradient. Sodium ions then diffuse down that concentration gradient into the lumen by facilitated diffusion. But this carrier protein is a special carrier protein that can co-transport glucose molecule in. This is represented here by S. Now this can be glucose, or it can be an amino acid molecule, or indeed a vitamin molecule. Obviously there are different carrier proteins for these different molecules. So they will get co-transported into the epithelial cell. And then because we then increase the concentration of glucose or amino acids within the epithelial cell, they will then diffuse across into the bloodstream via facilitated diffusion. Always remember, when you are discussing or answering a question about transport, you need to think about these four things in your answer. What is moving? Is it glucose? Is it amino acids? Where is it going from or to? Be specific. From the lumen into the epithelial cell. What's the concentration grading? It's going from high to low. And how is it moving? Is it going by facilitated diffusion, active transport, or co-transport?